good afternoon to all of you. All the citizens and the students of Bangalore. It's a very important meeting that having today, and thanks for gathering and on a Tuesday. And because of the COVID pandemic, other than the professional biologists, many people have come to know about what is PCR, RT-PCR, what is an RNA virus versus a DNA virus, and so forth. In general, I'm sure all of you are familiar with the central dogma of molecular biology, where genetic information is coded and encoded. DNA, it gets transcribed to RNA first, and then get translated to proteins, which are the real workhorses in the cell. But RNA is not only as an intermediate, it also has a lot of other catalytic functions. RNA is also being proposed to be the original genetic material before the cellular form of life evolved, which we are what exists today in the early days of the origin of life. And in that context, RNA biology is taking more importance nowadays, and better tools are available, better techniques are available in addition to the insight that we have on RNA itself. And many viruses are using RNA as a genetic material directly, but still in the host, they first get converted to DNA before they make multiple copies are made. That RT-PCR reaction that we all use for diagnosis is mostly because you first convert RNA to DNA and then you look for what is in that genetic material. Is it the SARS-CoV-2 or some other virus, or within the SARS-CoV-2 with new variant by sequencing we used to identify. And also when vaccine was developed, RNA was used as a vehicle to for the vaccine, and that made a breakthrough and helped us contain the infection spread. And RNA being also proposed, and already people are working on very different ways of for both diagnosis and therapeutics. So in that context, it's very apt that we organized this meeting in the year 2023, when just post-COVID, when RNA is taking so much prominence, and also this year the Nobel Prize in Medicine and Physiology was given to the people who developed RNA technology for therapeutics as well as for vaccine. So without much ado, and I request them to take over and conduct this meeting. And we have fantastic speakers, then we're followed by panel discussion. Please feel free to interact with them, try to understand more about it. Those who want to enter into this profession, and feel free to contact all of us. Will basically facilitate people who want to get into it. Thank you very much, and I'll hand over to Roopsi now.
I was just welcoming everyone to be here on a Tuesday evening. I'm Roopsi, part of Science Communication Outreach Office at National Center for Biological Sciences. Some of you already know NCBS, some of you who don't. We are based in Bangalore and one of the centers for Tata Institute of Fundamental Research, an autonomous institute under Department of Atomic Energy, Government of India. We are an institute dedicated to fundamental research in field of biology, encompassing a wide range of spectrum of scales in terms of time, complexity, and size. We have 4 institutions on a campus: NCBS, InSTEM, which is Institute for Stem Cell Science and Regenerative Medicine; CCAMP, which is Center for Cellular and Molecular Platforms; and Tata Institute of Genetics and Society, which is also called TINGS. We are excited for today's public event because we just couldn't find a better time to do this. Earlier this year, Katalin Karikó and Weissman were jointly awarded a Nobel Prize for Physiology and Medicine for their discoveries that enabled the creation of mRNA vaccine, including some of which that protected everyone against COVID-19. We think it is the perfect time to now also delve into India's journey of COVID-19 vaccines, and also explore the incredible platform of mRNA-based platforms that will be the future of vaccines and therapeutics. Without taking a lot more time, we will kick-start the evening with Dr. Sanjay Singh's talk, which will be followed by another talk by Dr. Aarti Ramesh, and followed by a panel discussion on past, present, and future of mRNA-based platforms in vaccines and therapeutics. Before we move on to Dr. Sanjay Singh's talk, I will give you a quick introduction about Dr. Singh. Dr. Singh is the founder and chief executive officer of Genova Biopharmaceuticals Limited based in Pune, which is a biotechnology company dedicated to the development and commercialization of safe, efficacious, and affordable biotherapeutics. Right from the onset, he was focused on innovations in biomanufacturing, technologies that culminated in commercialization of 7 life-saving biotherapeutics in the cardiovascular, neurology, nephrology, and oncology market. He has led Genova to develop an mRNA vaccine and establish India's mRNA-based vaccine platform to combat current and future pandemic-like outbreaks. Dr. Singh.
Thank you for this invitation to join this August gathering and give my piece of what the area Genova is focusing and where looks like the current possibility is. So without wasting time, I would like to go directly to my slide. And I just wanted to make sure that I'm visible well. Is the slide is okay on the, okay, I can see it.
You can make it a full screen mode.
Yeah.
Yeah, perfect.
Thank you. So just introducing the subject, and it is a very nice topic to discuss about demystifying the vaccines, the evolution of mRNA-based platform in vaccines and therapeutics. So going forward on my talk, I would like to, as Dr. Sidara said, in a very basic format, I would like to say that the information is stored in the form of DNA. And this storage is basically the instructions, and DNA is such a solid molecule, it can stay for thousands of years without any disturbances. But DNA is the only work as a storage, is transform its message or transmit its message in form of mRNA, which now we say this is a software system for the message. And this mRNA finally get applicable in terms of when the proteins are formed, which do the function. So this is the way we say that the central dogma operates. And you know the protein is the basically reason for most of the function or all kind of functions. If we go further into this direction, if you see this year Nobel Prize, which was given to Katalin Karikó and Weissman, and I remember the day the Nobel Prize was announced, somewhere very early in the morning, I saw the very afternoon, I saw the message from Dr. Sasidara. And I told him that Weissman was very thankful to 5 people who has taken the product to the market, and I was one of them. So this is a great service, and the Nobel Prize came so fast because its applicability was demonstrated in 2021-22, and within a year or so, the Nobel Prize was given. Basically, what they have been demonstrated, the leading work, where they have used base-modified templates for the mRNA, and which, in their view, is reduce the inflammatory responses. But overall, the vaccine has been made based on the technology which they demonstrated a couple of years back, but it has finally reached the market. In the term, I will say the reach to the market is, if you see the lower one, and this is from the finance side, only one of the vaccines, which was made by BioNTech Pfizer, sales was around $37 billion. And the second vaccine, which was Spike vaccines, was $18 billion. So around $55 billion revenue came with this technology in no time. So yes, scientific interests are very, very important. They solve very big problems. And in this case, they have saved the world, this technology, so quickly. And in terms of financial outcomes, you can see these are the biggest outcome for any man-made product, in such a short lifetime. Going back to, because this talk I have put it like just like a very general talk, so it could be very helpful for everybody to understand. So yes, the mRNA was discovered somewhere in 1961, and if mRNA has to be protected or given in any format, it was first demonstrated in a tube in 1969 that you can make the protein inside the tube through in vitro translation of mRNA. In the meantime, the first liposome was also produced somewhere in 1965. Why I'm saying it? Because these go hand to hand together. In 1971, liposome was first used to deliver a drug. Then also liposome was used to deliver some initial vaccines. And in 1975, a new revolution came that there's a cap is required on the upfront side of the mRNA, which protect mRNA from degradation. And finally, in 1978, it was a proven thought process that in vitro translation of mRNA is very much feasible. So I would like to say that you have to keep three facts. One is 1961, second is 1971, in vitro translation, discovery of these caps. There are many development, but I have taken few of the which was the major one. Now, again, I have just pointed out liposome and delivery of a vaccine through liposome is very, very important, because my talk will be somewhat bring a lot of focus on this particular topic. Further, starting from 1983, cap analog was commercially synthesized. Then you can synthesize the mRNA in the lab, T7 polymerase you can produce, and you can start going forward into different possibilities. And the most important thing came out in 1977. A first company was founded based on mRNA, and then very famous company which is CureVac came in 2000. So this is the side of commercial side started going into it. This is the third one. This is a quite busy slide, but in this busy slide, it realized that it has to be delivered in a nanoparticle. So 4 components of how the nanoparticle has to be made, or liposome has to be made. BioNTech founded in 2003, and then many things started, and basically injecting the mRNA into the animal, into the human, all these started. And first clinical trial was mRNA vaccine was in somewhere in for rabies in 2013. This is also I'm telling from because we will follow this point also. And then CRISPR-Cas9 and all these things were very busy. If you see things started came, but basically it was the end of 2020 when the first COVID vaccine was given the emergency authorization, and now from there this whole field has opened. And where the today talk is also focused is mRNA-based therapeutics. Now, going a little bit into basics, the 2 types of base RNA are available where the technology has been used. One is vaccine immunogen, where the conventional sequence you can put it here in terms of plasmid. You can have 5' UTR, 3' UTR, a poly A tail, and basically a cap structure you have to put. And this was used in Pfizer and Moderna's both vaccine. But as a unconventional, I moved ahead. I have not used the conventional mRNA platform. We decided to use the self-amplifying mRNA platform. In self-amplifying mRNA platform, we put a machinery again in front of the immunogen sequence, and which makes multiple copy of it. And we were knowing this is important because otherwise mRNA is given within 8 to 10 hours, it's produced a protein and gone. In this case, your immunogen is surviving somehow for 15 to 20 days. And this experimentally others have proven, and also we have also proven. What it gives us the opportunity to mature the B cells, so we may have a better memory response as compared to non-replicating mRNA platform. So these are this is the standard slides, but we present that first generation vaccine came, and which is like whole organism and polio, hepatitis, rabies, influenza, yellow fever, measles. Many vaccines came from this. They started from Professor Jenner, maybe like 80 to 90 years ago. And this platform is still continue. And one of our vaccines in the country was made, Covaxin was on this platform. The second platform is since 1965, 70s, recombinant DNA technology started. And slowly, it has also converted into vaccine, where you can see the sequence, which is basically have a clinical correlates of protection. And those antigens you can produce recombinantly, and one of the most successful vaccines into this area, few of them I'm mentioning, is seasonal influenza vaccine, hepatitis B, and HPV, very, very important. And this is the way the second generation started. And we say the third generation, which came on DNA vaccine, started around like 20 years back. And here, one of the vaccines which was emergency use authorization was Ebola vaccine, which was made on adenovirus not replicating DNA platform. And the next vaccine which got approval, it was developed by Oxford in collaboration with AstraZeneca, and in India it was manufactured by Serum Institute. So this is the vaccine which came from generation third, and this is what we used to say generation 4, mRNA-based. This is a total synthetic platform. Most important thing, we don't need a fermenter, we don't need a bioreactor. This is total synthetic-based platform. You have a plasmid, and you in vitro translation, you convert into the mRNA, and then mRNA can be given. And here, first vaccine was brought by Moderna and Pfizer. The second vaccine was from Moderna and NIH. And we were the first outside US and third company which got the emergency use authorization of our vaccines. Here, this is an important fact to see that process economics was very costly, but time of manufacturing can be reduced very, very little. This is what, with time, this technology is giving the promise for this. Now, going back into it, and where you see the Nobel Prize is also dedicated, that basically this is the platform which is open replicating frame. You can have 5' UTR, 3' UTR, poly-A tail, and different kind of cap structure, cap 0, cap 1, cap 2 now just has come a few weeks back. And here in this open reading frame, what was proposed by Weissman and Katalin both, that you can use these modified nucleotide sequences. And when CureVac went to make their vaccine, they have not used these modified sequences. Though when we've gone ahead, and somehow this CureVac vaccine has not reached to the market. These both vaccine, Moderna and BioNTech, Pfizer has gone, and this is what we use. We have also not used these modified, because when we go into the system, first cycle is all right, modified bases are used, but when again negative strand has been made and then resynthesis started in body, there is no nucleoside modified versions are present. Non-modified nucleoside are present, so we have not used it, and still because of our thought process, we succeeded very well. And a lot of work today is going on this, how to develop modified poly-A tail, what's the length of this poly-A tail. A lot of research is going on 3 UTR, a lot of research is going on 5' UTR. In similar fashion, some research is going on in the area, how you design the new capping. So basically, why we decided, we thought that very small quantity of self-amplifying mRNA we can give, and it will reduce the burden of raw material, reduce the burden of supply material, and it should be better immunogenic. This is what the thought process we had. Now, as I talk about the liposome, this is liposome, and with what we say, this is our version. So both the here we tried to put our mRNA on the surface of liposome. We have not put it inside. And the reason for this was that it is very easy to manufacture, because when we make this membrane vesicles and forcefully through microfluidization through the mRNA, there is a lot of losses of mRNA. So you have to use, recoveries are not more than 70-75%. In our case, the recovery is more than 95 to 96%. This is one of the major reasons. And also producing that nanofilled mRNA liposome is not near homogeneity. And here we demonstrated that we produce these cells very close to homogeneity. This is the part one. The second, we are not using PEG, because PEG has its own inherent some problems, and we have not used PEG in our liposome. So this is the liposome technology. And here there is no water pockets are there where the mRNA is running around. And why it is important, I will go and present in the next slide. So this was one of the biggest problems, the ultra-low temperature for these both vaccines, either Moderna or Pfizer. And when we had interaction with the Honorable Prime Minister, with our DBD Secretary and all over the team, they were thinking to procure the mRNA vaccine from Pfizer and Moderna. But the problem was that we are not equipped as a nationwide to have the cold storage system, which is ultra low. So the only solution was, can we make mRNA vaccine stable at 2 to 8 degrees centigrade? Honorable Prime Minister asked one-to-one, and the way he see into eyes, that is it possible? And my answer was, yes, it is possible. How much time it will take, we do not know. But this I assure, if few groups in the world can solve this problem, Genoa will be one of them. Going into logistics of into it, if you see that this is one type of liposome, this is another type of liposome, this is a third type of liposome. But here is the middle one, which is basically has been used for delivery of this mRNA vaccine. So you can see small pockets in which mRNA is placed. And this is, we were looking into it, and fortunately at the same time, a group was publishing these papers. And if you amplify it, you can see these are the water channels where mRNA is there. So there is a presence of water channel. And now this is here the simple chemistry, but for us, it took time to think about it, that why DNA is so stable, because this is the difference is hydroxy bond, hydroxy group over there. And we said that this is one of the one stupid atom which make mRNA so unstable. By design, by nature, yes, mRNA is the only messenger. It's to transmit its message and finish it off, by which the God has, I think, designed it, or the system has, evolution has come in this fashion. But it is also a problem when we think mRNA as a vaccine or mRNA as a storage material. And when you go into deep detail, you can see the presence of H2O. It reacts with the hydroxyl group in the base, and basically it goes and hit the phosphodiester bond, and finally bases are broken down. And this was such a simple chemistry, and we realized, yes, water is a culprit. So our design for the liposome was very critical, that we can remove the water. So what we did in the first prototype vaccine, which was approved, we lyophilized the vaccine and water was removed. And when we tested it in comparison with the -80 frozen vaccine, there was no difference into the titer value. Even when we tested it for the potency, we found that if you see this, the second one, here the response even came very quickly from the first injection, and then it was almost equal to the second injection in the booster one. And we have this data now almost, this is a 6 month, but we have 12-month data available. 18-month data is also available, that this vaccine is even stable at 30 degrees centigrade. And we feel very proud that somehow we have solved this problem. And because if these vaccines has to be globally deployed, only few countries in the world who can take care of -70 logistics and supply chain system. This was the second part, which was a problem for us, that raw materials, all the raw materials we have to take, these raw material has to come from outside. Either you take T7 polymerase, pyrophosphatase, RNase inhibitor, DNase, capping enzymes, everything. And a lot of lead time and prices, exorbitant prices. So only one solution to do backward integration. And today Genoa has done most of these backward integration in-house. And we can say we are more than 90% self-reliant, either by us made these things in-house, or we have used our partners to make this. Like even lipid, which was the most important component for the vaccine, very costly component, has been produced here internally in the country, and we can produce in Darmstadt. Now, this is one very important factor that in future pandemics, are there the need? We have broken this whole process into 4 parts. The part 1 is we can produce plasmid and store it as much as you want. We can produce mRNA, we can store it. We can produce these nanolipids and we can store it. This is the advantage of non-entrapment. And then we can mix together and we can go ahead lyophilize. Few people, most of which you are aware of, that WHO was trying to create mRNA hub. And initially I did not participate because I wanted that how I should transfer the technology into these hubs. Now, plasmid can be at some places, mRNA can be some places, CLNE can be some places. And one of the places which we decide to deliver the vaccine in very small quantity in kg, this material can be shipped and which can be finally lyophilized and produced in those countries. So now we are working actively with the WHO to establish these hubs because Moderna and Pfizer didn't participate into the program. So first was our vaccine, which was Gemcovac 19 was approved. And then we again developed the booster version, and this is the only booster available in the country. We developed Gemcovac Om, which is Omicron version, and which has also gone through the full trial, and now both the vaccines are approved. And this vaccine was launched by our Science and Technology Minister and on June 24th, and now both vaccines are available on the CoWIN app, and it can be ordered, it can be used because hospitals order from this one. So this is a very little insight I have tried to give it. It's a very complex work because I want to thank more than 7,000 volunteers who participated in the trial. The trial was all across the country. First trial was around 30 places, second trial was 22 places. But this was the tough journey we have gone through. And in terms this, both the patent we had went into, and one of the patent was how to wrap the mRNA on the surface of liposome. That was a very, very difficult process, but we finally succeeded. And we can produce from 200 microliter to 200 liters or 2,000 liters, these solutions with algorithms. So that was the first algorithm we filed as a patent. The second was algorithm was that how to lyophilize that, and when you bring back into the water, no mRNA damage or no liposomal damage happens. So that these are the two art. We don't say technology, we say two arts we developed, and both the patents are there. Few publications are in the queue, and soon you will see the publication coming from it. So where we are with these technologies and what we want to take it forward. So first thing is still we are trying to do some research on the mRNA platform, and here I will say the scientific community or academic community to look into these, and we are very open and transparent. Can we work together? Because all the work cannot be done in the boundaries of Genoa. So one is mRNA editing. So this is another thing which we have few partners in the country and outside also, and where we can see that in which case self-amplifying is going forward, non-self-amplifying can go forward. And just for information, BioNTech has just started a vaccine trial using self-amplifying vaccine. Simplifying platform. How we can further innovate into the in vitro translation mechanism, how we can go further and develop certain nanolipid preparations. So here, few of the groups are working in the country, like CMC Bangalore. They are working on some new novel lipid formulations, we are working with them, and how we can further improve our vaccine formulation. Because nanolipid is finally, if we have to deliver targeted, just like one of the new area which is coming up very fast, maybe whole CAR-T technology is going to convert into mRNA-based CAR-T technology. So you don't have to isolate the T cells and grow them T cells, and then you have to transfect them with your particular vector, and then you have to inject it again. Here you can have your sequence and you can target the T cells and you can hit it by single injection. So these are the very, very, what you call, frontiers are open for us. And then also backward integration, can be one of the leaders in the world to do this.
Dr. Singh?
And then,
Hello?
yes.
Hi, we have about 5
I can
minutes.
hear you. Yeah,
we
I'm
have
just
about-
on the close of this.
Thank you, sir. Yeah.
And then going back on what we are doing out of it. The first vaccine went, the variant vaccine went. We are also working on third generation. And there are a few vaccines which we are trying to improve, which is like shingles, dengue vaccine. We are also have interest in malaria. But few of the novel vaccine which we are hitting, and one of them is tuberculosis, because our country believes that we can eradicate tuberculosis by 2025. A very daunting task, but is participating into that program. That's how it goes. Similarly, on a bigger vision, if we see all these two things together, what we want to deliver, we want to have India should be placed for mRNA gigafactory. We should create national and regional stockpiling centers. I said to you that through mRNA hubs kind of thing, we are open for technology transfer, and that which will give the deployability ability of this our thermostable formulation. So basically, this is a very unique slide. We love to present it. That this is a disease agnostic platform. On left side, all the components which are required, they are common for any vaccine or technology you want to develop it. You only have to change the sequence. You have to put it together and finally mix it, and you will have the vaccine. For this, facilities are same, analysis are same, training is same, process is same. And we have demonstrated 2 vaccines which got the approvals. So now we have just partnered with CEPI, and they are focusing that can we improve our platform further for different diseases. So as you said about the rabies, we are developing through this disease X technology platform for the rabies. And today what we say that can we reach a goal of 100 days is possible with simplified and clear regulatory pathways. We are working with the government for can we develop a vaccine using this technology within 100 days. So I want to say these are the 3 things which we have worked out: pandemic ready platform, ultra low temperature problem solved, and acute shortage of key raw material. As an industry partner, we have solved these things. So thank you very much. And just to say that last week, one of our scientists was in a meeting in Berlin, and they were presenting next to each other with one of our Nobel laureates who got the Nobel Prize. So thank you very much from my side.
Thank you.
So now we are open for the question.
Thank you so much, Dr. Singh. We can maybe take a question or 2. Yeah.
Yes, go ahead.
Quick questions. One is what is the fundamental difference in terms of cost and efficacy between the DNA and RNA-based vaccines? That's one question. Second is, at least I have taken this COVID shield, both the booster and the whole thing. Now, is it possible to shift to mRNA-based vaccine, or is the stream need to be continued in terms of technology? And third is that, you mentioned about other vaccines being developed, including rabies and few others, using these technologies. So what is the timeline? And specifically for rabies, I'd like to know, will it be cheaper, better, less number of vaccines, any other advantage people will get? Yeah, those are 3 questions.
Okay. So first question, that how the difference between DNA and mRNA vaccines. We have not done any head-to-head comparison between DNA and mRNA vaccines, but mRNA vaccines has been proven as efficacious as any possible vaccine, because this based on correlates of protection, and looks like correlates of protections are very good. In non-replicating vaccines, somehow the titer has gone faster in 4 to 6 months. And in the meantime, the new variants has also come, so that very quickly you can make the new variant and you can go ahead. So I think overall, for rapid deployment and efficacy point of view, I don't see there is any negativity in this platform. But I feel mRNA vaccine manufacturing is much more easier, quantity-wise, delivery-wise, as compared to DNA vaccine. This is what, with my own experience, I can say. The second question you asked about if somebody has taken AstraZeneca vaccine, Covishield, whether they can shift to mRNA vaccine or not. So Canada, Europe has started using this vaccine first produced by AstraZeneca, but as soon as mRNA came, most of the people has switched to the mRNA vaccine even for the second dose. And the boosting has also been, and WHO has given guidelines that mRNA vaccine can be used who has taken from other platforms vaccine, because finally you are going to raise the inside the memory response itself. So there is no problem, and guidelines are there. And the whole Europe, the whole Canada, especially they moved into the where initially AstraZeneca vaccine was there. Third, about the rabies and other, yes, we have just started recently, 20-25 days back, we have touched the rabies program. And rabies program we have touched because of the reason there is a shortage of rabies vaccine. And hopefully this new platform is going to give a better vaccine in terms of safety, in terms of delivery, and in terms of efficacy. But we are going to do head-and-head trial, and hopefully in 6 to 8 months, we will come back to you to give the results that how these results are going to come. And we are very positive from the preclinical works. It is as good or better than the existing vaccine which are in the market. Thank you.
Hi, my name is Neha Panko. I have a question about how you decide which vaccines you're going to take on to your mRNA platform next. So is there a process in that decision making? Is there a business model, or what is it driven by?
Yes, it's a combination of all, but we feel what is the need base. So need base, I feel the dengue and tuberculosis vaccines are most needed, because maximum people have died in last couple of years. Other than COVID is the tuberculosis. So that is one we want to take the challenge. And India harbor around 45% of the tuberculosis cases in the world. The second problem with tuberculosis is multi-drug resistant and XDR, which is one step ahead. So I think tuberculosis vaccine on the target. And you might have seen in this year that dengue has touched almost every home in the country. So these are the two priorities areas we have made a decision. And rabies is the one thing which we are working with CEPI. So these are the three vaccines which are going forward. But certainly there is a business model, because we basically trying to focus more on the personalized cancer vaccine. That is also one of the area where we want to go using new antigens platform.
Thank you. Maybe we can take one last question.
Should I stand up or should I sit down and ask? Yeah. Doctor, I came in between. I have not heard the complete meeting. But what I came to know that government is slowly planning to shift to mRNA platform. So, yeah, can you hear? Yeah, I'm Smita, sir.
I can hear.
Yeah. From the conversation, from the whatever slides I came to know, I came to know that the slowly government is planning to shift to, it has already approved some mRNA vaccines, mRNA-based platform vaccines. I just want to know, what are the, what are the what precautions or what kind of what department has been assigned which takes care of any adverse effect in future? Because from the experience of US and Canada and Australia, since mRNA vaccines were introduced there, we are seeing millions of deaths, lot of adverse reactions, all kinds of diseases have, and turbo cancers have grown. And basically, I am associated with Awaken India movement, so we know what all havoc it has caused. So I just want to know if it should not happen with India, that we see in every house 2 or 3 patients. So what kind of precautions or what have steps have you taken in case? There is hardly government listens to any side effects. We have seen that in last 2 years. There are some people who are guiding the government and they are just trying to suppress all adverse events. So just want to know, like mRNA, it will create a havoc. You may not agree because it's a business point of view, but as an Indian, I want to know, sir, what kind of, in case of adverse reactions, how are you going to handle it? And I'm sure it will happen as per the experience in last 3 years in European and US, sir. Thank you.
Okay, so thank you. I'm as Indian as you are. I'm a scientist first and a businessman second. These approvals are based on many factors, many committees, many reviews, thousands and thousands of patients in the human clinical trial. On the basis of that, it goes on. Yes, a lot of news is around, comes and goes, but we have to go through the scientific evidence, that what are the scientific evidence which has been found. And I think US FDA is one of the toughest regulatory authorities in the world. Each and every adverse event has to be reported. We have done around 7,000 patient clinical trial, and one of the comparator arm, we saw the evidence, and for that our clinical trial was stopped for a couple of weeks. We presented all the results that this is from the comparator arm. So at present, we do not see anything which is, because you have to see from benefit versus risk ratio, all that, there's none of medicine, even if you take paracetamol, it has some side effects. Nothing, no medicine can have without 0 side effect. That we have to accept. But mRNA is a basically natural product. So how the biologics has come into the world, if you are having diabetes, you have to take insulin, because it is the safest part, because it is produced by your own body. The same way mRNA is produced by your own body. So there is nothing like, which is otherwise, I think 3.8 billion people have been given mRNA vaccine. 3.8 billion doses has been given. But certainly I agree with you, there should be a watch list. Many, many years it will need to be followed, that how this area is going to develop. And I would like to thank DVT who believed into mRNA so early and also given part funding to us. And this is not only one department, but many departments which participate into it, say the review. But again, yes, the data, the science and evidence are going to find the ways, which is what is truth, what is not truth. So I don't want to reduce your concern, but certainly it is watchful, and we are the most vulnerable, so we watch it very carefully. Thank you. And just for your knowledge, both the vaccine I took first when they were made. Nothing more bigger evidence we can give, because we are not sitting on the sidelines and watching the game. We are part into it, and I have taken both the vaccine first.
Thank you so much, Dr. Singh. Now we have Dr. Aarti Ramesh. She is currently Principal Scientist at Tata Institute of Genetics and Society and was formerly a faculty at National Center for Biological Sciences. Her research group explores the biology of RNA in microorganisms, how RNA sensors help microbes sense their environment, and the role these RNAs play in making microbes resistant to antibiotics. I would like to welcome Dr. Aarti Ramesh.
So good evening and thank you all for joining. I am Aarti Ramesh. As mentioned, I am a scientist at the Tata Institute for Genetics and Society, right here in Bangalore. I thought in the next 20 odd minutes, I'll walk you through this world of RNA, a molecule that I am deeply fascinated by, and I hope by the end of this whole session, you all are also equally fascinated by it. We'll start with something which we're all very familiar with, which is our body, right? So our body is made of cells, and there are cells everywhere. There are red, cells in the blood. I need a pointer. Is this the only pointer to use? Yeah. So there are cells in the blood. There are cells in our nervous system, cells in our lymphatic system, etc. And what's important to know is that every cell is essentially a bag of proteins, right? In the many trillions of cells that are in our body, there are many thousands to a million proteins in each cell. And what keeps these cells, or what dictates how these cells look, where they go, how they divide, how they grow, etc., is all decided by the proteins in that cell. So the form and function of a cell essentially comes from its proteins, right? But how do these proteins come about then, right? Proteins come about from the DNA. Some of this is going to be repetitive with Dr. Sanjay's talk, but RNA is so important and so interesting that it's a good thing said twice. You can hear some of this again. So anyway, the protein comes from DNA, and this DNA is essentially the code or the instruction sheet, which is the hereditary material. This is in fact kept in a nucleus, think of it as a safe compartment for the DNA, and it has many different chemical features which distinguish it from the protein, although it carries the code which then translates into the protein, right? And for every protein in the cell, there is a code that is mentioned in the DNA. Between the DNA and the protein, scientists figured out in the early 60s that there is an intermediate molecule, and this is called the messenger RNA. It's called the messenger RNA because it takes the message from the DNA, and eventually it stays true to the code that the DNA has. So it actually has the same code, right? But then this code is moved from this protected environment, which is the nucleus, taken out to the rest of the cell where all the machinery exists to make the different proteins, right? So this raises a question: why do you need a DNA and an RNA when both of them essentially have the same code, right? In fact, viruses, if you think of COVID, don't even have a DNA, they have only RNA, right? But you need these two because there are some very basic fundamental differences in the way the chemistry of these two molecules are. Very subtle differences, but that makes a huge difference to biology, okay? So one is the component molecules are slightly different. You'll have to look at all the atoms to know the difference, but just believe me, they're slightly different. And what these differences do is that they result in an RNA which is largely single-stranded, and a DNA which is largely double-stranded. Now, this makes a difference to biology because the single-stranded molecule, which is the RNA, is now very flexible and is able to take up all sorts of shapes. These gray things are RNA, right? Something which the DNA can't do. It's protected, it's hereditary, but it's largely very stable as a structure. RNA, on the other hand, is exported. It is not very stable, and it can do a lot of fascinating shapes which allow it to do a lot of biology, a detail which I'm happy to talk about later if there's time. What's also important is that difference that Dr. Sanjay pointed out, where there is a single oxygen which is extra in the RNA compared to the DNA, and that extra oxygen makes a huge difference because it makes RNA very, very labile, which means that in a cell, although the information or the code is present both in DNA and the RNA, you can still get DNA from fossils. You can't get RNA from them because the half-life of RNA, or the time that an RNA spends in a cell, is just a few hours, whereas a DNA will last for eons, right? And that has made a huge difference to biology, that oxygen atom, right? So what early science figured out, maybe in the 60s with the discovery of messenger RNA, is that, of course, RNA is required to convert that DNA message into protein, right? But over the years, we have found, scientists have found that it does a lot more. This flexibility, the transient nature of this molecule has been used by cells for many, many different parts of biology. This is, of course, the mRNA itself, right, which is the code which binds to, which recruits a protein. So this RNA strand in the cell can bring a lot of proteins towards it through some physical and chemical attractions. And in fact, that is what results in the making of a new protein chain. So every protein in the cell is made from an mRNA that is in the cell. But RNAs don't do only that. Apart from carrying the message, they even form this very machine. So the very machine that makes protein from these mRNAs, that itself is made of RNA. And there are many subtle details like this, where in the cell, RNA is used for much more than just transferring the code from DNA to protein. Right? This is generally the biology that was being studied of RNA, and now we know a lot more about what RNA do. But I thought I'll give you a slightly historical perspective which builds towards why vaccines and mRNA vaccines are particularly possible now. Right? So I'll talk only about that part of RNA science which has led us to the modern day vaccines. So about in the 60s, the mRNA was discovered. And then in the 70s, what people were trying to do was to see if this is now a molecule which can make protein, how about I make an RNA of my own and get it to make a protein of my choice? That is what drove the research, I presume, in the 70s. And what people did was, let's get the RNA out of the cell, right? The RNA was identified in the cell as a messenger RNA. So a lot of efforts throughout the 70s went in trying to bring a cellular RNA out of the cell. So people used all sorts of chemical and physical techniques, crush the cell, extract it with this solvent, that solvent, etc. Details which we don't need to go into right now. But they tried to bring out intact messenger RNAs from the cell. And a lot of efforts went into that. So successfully, mRNA could be isolated from certain kinds of cells. And when isolated, people tried to inject it back into a living egg to see what might happen. Then people figured out that when the mRNA was extracted, they started studying what's in this mRNA now, right? So a lot of chemistry led to the identification that mRNA, if that's the blue thing, which makes protein, has a cap at one end and a tail at the other end, right? And this discovery became very important because not only did scientists find that there is a cap and a tail, they also found that without the cap, the mRNA cannot make protein, right? This is fundamentally important to the modern day vaccines, which I'll hopefully explain to you how. So they found that there are caps and there are little bits of these frills and fancies in messenger RNAs, which is what tells a cell which RNA is to be made into protein and which RNA is to be left alone. Because there's a lot of RNA in the cell which also doesn't become protein. So the cell uses the cap and the tail to know which RNAs need to be converted into protein. So this discovery was very important. People also found, very interestingly, that viruses, right, probably the most clever creatures, right, have enzymes that can add a cap to an RNA of their own. Now this was important because if I want to make an RNA in a tube myself, what am I going to do? I will be able to put this part together because I know which protein I want to make. How am I going to get a cap to be added to that, right? This cap is usually added only inside a cell. But the discovery that viruses have enzymes that can take a host's cap, another organism's cap, and add it to their mRNAs became fundamentally important to what we can do in a test tube. Of course, people also isolated mRNA from one kind of cell, and then instead of injecting it into an egg, started injecting it into other cells to see if one can isolate an mRNA and then put it into a cell where it was otherwise not going to be found. And this is the kind of research that happened, which was important to know that you can actually bring an mRNA out of the cell, what all is it going to have, and then put it back to see if this is going to be functional when put back into a live cell. And after that, in the next decade, people started working on making this from scratch, right? So what they did earlier was to take a living mRNA from a living cell and then put it back. And this was a different idea. This was like, by this time in the 80s, people knew a lot about where RNA came from, what kind of a sequence could produce RNA, and what kind of an RNA sequence would produce protein. So then, and also DNA synthesis was now possible, right, in a tube. So once a DNA could be synthesized in the tube, this whole thing, the whole reaction of DNA to RNA to protein could then be made in vitro, which means inside a test tube, right? So now if one simply knows what is the sequence of the protein I want, it could be anything. I could create it from my head if I wanted to, or it could be a protein whose sequence I already know, something very important, the protein in the lens of my eye, for example, right? If I know that sequence, then I can have a DNA of that sequence, put in all the goodies in a tube, which convert the enzymes that convert DNA into RNA, and I would get RNA, right? And so this is the research that was very important, which allowed us to start making RNA in a test tube. So what did people do? The details on the slide are not particularly important, but this goes to show that people could, in the 80s itself, make RNA in a tube, add the caps, etc. to it, and then simply add it to a lysed cell, right? Simply give it all the goodies that are required to convert that RNA into protein. I think this was groundbreaking in some sense, because now one can make something completely in a test tube, an RNA, and it could be translated into a protein in that test tube, right? So just to show you where time stood, so this is Sidney Brenner in 1961 discovered the mRNA, and after that, like I showed you, through the 70s and 80s, people could start doing in vitro transcription, which is making RNA in a tube for a sequence of their own choice. And then there were these mRNAs were being injected into live organisms such as mice. So what will happen if you actually put an RNA? So far in the 80s, like I told you, people were injecting mRNAs into cells in a petri dish, right? Cells in a petri dish don't have this thing called the immune system, right? Bodies have immune systems. So by this time, when people had started to inject these mRNAs made in a tube into live organisms, they realized they had a problem to contend with. This problem was the immune system of the person you're injecting, or the mouse itself. And then, obviously, several years went into figuring out how do you now get past this immune system, because our bodies have, an immune system is meant to recognize foreign material, and any RNA injected into an immune system is going to be recognized as foreign material and taken care of. So that means that the RNA is never going to actually make it inside a cell, which means the modern day vaccine would have been impossible, right? So research went into figuring out what can you possibly inject as an RNA that, I mean, obviously the cell has its own RNA. So RNA fundamentally is not a problem for cells. It's only the RNA that you're injecting that is a problem for the cell. So they started comparing what are we injecting, how is that different chemically from the RNA that's already in the cells, and why is this not being recognized as a problem, but the one I'm injecting is a problem. Several types of RNAs were isolated from different species, injected back into mice to see what elicits an immune response. And what people found was that there is a fundamental difference in the kinds of RNA that bacteria and viruses have compared to the ones that they were encountering in humans or in mice, right? And that difference is that there are special protections on the RNAs that are recognized as self, okay? And these protections have different, they are basically, think of them, all of them are different chemical moieties, but they all are essentially decorating the entire RNA. So this is one type of RNA, this is another type of RNA, right? These squiggly lines and all these little colored boxes over here are the kinds of protections. So if I have an RNA which is made of 100 subunits, this kind of an RNA called a transfer RNA, 50 of them are going to have this protection on them. So they have all the features of an mRNA, all the chemistry of an mRNA, but on top of that chemistry, they have these little protective caps, if you will. Okay? So then it was hypothesized that maybe these caps, actually, they do many other things. Caps are important. These protections or modifications is what the technical term is. These modifications of the RNA are required for various types of biology. But what people could use it for is to now introduce these caps in the RNA that was made in a test tube and then inject back that in vitro artificial RNA and they found that the immune system was able to recognize this and take it up, right? This discovery of modifications on nuclear bases or on RNA became fundamentally important, because without this, you cannot inject anything and evade the immune system. Okay, so what happens now after you have injected? Will the RNA enter the cell? If it were to enter the cell, it will be just fine, right? Because now you've protected it with all these modifications. But there was another problem to contend with. This problem is that naked RNA can't enter the cell for various reasons. The cell is, the membrane, our cells, every cell has a membrane. This is meant to protect the cell. Otherwise, everything would simply be entering, right? So that's not how these have evolved. Membranes that surround the cell have evolved to prevent most molecules from entering. So there is a size restriction, there is a charge restriction, there is lots of physicochemical properties which prevent molecules from entering the cell. Only very select things can enter and leave a cell membrane. And RNA is not one of them, right? So in order to send an RNA into cells, one needs to surround it with a layer of fat, essentially, called lipids. And this is because these lipids can fuse with the lipids already forming the cell membrane. And various processes are able to take in a lipid vesicle full of RNA into the cell. So if you have an RNA made in vitro and which is now modified to prevent the immune reaction, you then coat it with a layer of lipids, right? It would enter the cell through some processes, biological processes, and that after entering the cell, the physicochemical nature of the inside of the cell is such that the RNA would be released. And once released, if you've designed it well and made it well in the tube, and it has a cap and a tail and everything that it needs to convert into a protein, this RNA would then be translated or made into a protein. That protein would then do the magic that you want it to do. Okay, so this is the general idea that evolved as people went through making RNA in a tube and trying to make protein out of it. Of course, I mean, the scientists studying lipids have many different types of lipids, and this research, as Dr. Sanjay pointed out, had been going on for decades, except that sometimes there's a certain things have to click in place. So the perfect combination of lipids, some that keep the RNA stable, some that fuse better with the membrane, some that make the particle size, whatever it's intended to be, that kind of research went hand in hand with the RNA research. And then when the two fields combined, it was possible to get these lipid nanoparticles, or LNPs, as you might heard of, which can be mixed with an RNA or an mRNA and then sent into a cell. Right? So just to take a sidetrack over here, now if all of this research had already built where you can make an RNA of your choice, make it code a protein of your choice, modify it as you want to evade the immune system, and now coat it with a bunch of lipids so that it can actually make it into the cell, what would you do with a system like this? What all can you imagine that can be done with a system like this, right? So, this is not about mRNAs right now. This is to show what happens when there is actually an infection. So when there is an infection, a disease-causing virus, it could be anything, but since it says spike over here, let's just think it's COVID, but it could be anything that's infecting you. The virus enters the body and then replicates inside the cells of the body, right? So these cells are special cells where the virus can replicate, and as the virus replicates, some proteins from the virus are now expressed outside this cell. This is a host cell. Host cell means a cell from the body which has been infected. So this is not a virus cell. So now a host cell starts to present proteins that which actually belong to the virus, right? Why this happens is because these cells are now recognized by the immune system of the body, right? And the immune system of the body now recognizes these cells and recognizes this new antigen, which is a part of the virus, which is now being exposed on the surface of these cells. And the immune system then gets triggered. Two kinds of things typically happen. One part of our immune system starts to produce these things called antibodies, which we've all probably heard of, right? And there are these B cells which start producing specific antibodies which can neutralize these antigens so that they don't have a harmful effect on the body. And then, of course, there are these killer cells, cytotoxic T cells, which can completely destroy the cell itself. So as to say that if there is no cell, then the virus inside it will also be destroyed, right? So this entire immunity kicks off. The problem is that we can't wait to be infected by a virus and for our immune system to take care of it, right? Because A, somebody may be immune compromised and may never reach this stage, right? In which case the virus will keep propagating and you'll be really sick. The other problem, which was seen particularly for something like COVID, is that after the infection, your immune system may go nuts. So if you start having such a severe immune response that now, the inflammation in your body itself has now become a problem for your own body, then you can't really wait to see how strong your immunity is and whether you're going to deal with this virus or not. I mean, some people were just fine after the infection and this whole thing happened, and some people had a problem either with too much virus, right, or with too much immune reaction. So in order to avoid this, one uses vaccines, right? And what a vaccine does, this is a little redundant, but it's interesting, so you can hear this again. What a vaccine does is it sort of gives a teaser to the body that this is not a real infection, but something like this can come your way. So you be prepared. That's the concept of a vaccine, right? Traditionally, people would take the whole virus, inactivate it, make it a little weaker and a little dead, and then inject that, and your body would make an immune response to it, and then those antibodies would last so that if you actually got an infection later, you already have a little bit of a store and a little memory. Your immunity already has a memory. After that, people, as more knowledge grew, one could identify what are the proteins in this virus that are important for creating this immune response, purify only those, and sometimes use those as a protein-based vaccine, right? Then there are these viral vectors, which some of us took, where you now take this protein, which you know is required to cause an immune reaction, put that into a virus, but now it's not a dead virus of a problem virus, but it's a virus which is a friendly virus, a virus for which we don't think has a big effect on our body, and that is called a viral vector. And that is now the latest, has now become an RNA vaccine, where because we know the code of this protein that we want to express, the protein of the virus that is going to elicit a huge response in our cells, we can encode that protein or a part of that protein in the code, send that code, which is the RNA, into the cell with all these modifications and all of these lipids, etc. And because that technology now exists, the RNA can be sent in, but the result of all of these is to produce antibodies, right? And of course, we have a panel discussion after this, but there are different advantages and disadvantages to each of them. So I don't believe that one is going to replace the other, but I do believe that now more options are possible, so one will be able to use the pros of these different types of vaccines. So, yeah. This is a little redundant, but this just goes to show that it took, from the time that the mRNAs were discovered, it took about the early 2000s when people actually started doing therapy based on mRNA vaccines. So you could start doing this injection with whole mRNAs in subjects that many decades later. But I wanted to add over here that now by 2013 to 2017, several trials were already going on, right, for mRNA vaccines. There were already trials going on. So what happened that we saw the COVID vaccine, the mRNA vaccine, as the very first one that got rolled out? So this is something that we'll probably discuss again at the panel, with the panel also. But I believe what happened really, that the pandemic did something that had not happened until the 2017 trials of the mRNA vaccines, right? So this, I think the scientists were doing this systematically and had reached a point till the 2017 where you could actually have an mRNA vaccine and do trials. However, the pandemic brought a sense of urgency and a sense of collapse and collaboration, which made this, made it possible to roll out a real vaccine, get an approval, start injecting people, right? Because that sense of urgency was not there previous to this. There was already a Zika mRNA vaccine which was in monkey trials. There were other cancer immunotherapies that were already on in trials. But what a vaccine needs is more than just the science. The science has to be solid. You have to know all of these things about the mRNAs and what to inject and what not to. But then finally, when you wanted to show results at a level that was seen by the pandemic, you needed more. And what you needed was that you needed all of the science behind making this. You needed, of course, the protein sequence for COVID, etc., which could rapidly come about because the technology to sequence a DNA was now finally super fast. You had this ability, again, the science was there, but to produce it and package it in a way that it could be used, the materials people, the people who actually manufacture these at scales which are relevant, all of these were missing before, or at least that sense of urgency was not there probably before the COVID thing. And then after this vaccine, of course, this is what happens inside the cells, like we have gone through. So I wanted to leave you with the thought that that pandemic, you know, the one thing that happened for the RNA world because of the pandemic is that it really accelerated the mRNA technology. Some of the advantages, and this is again going to come up in the panel discussion, so we'll keep it brief, is that you know, somebody had asked a question about DNA versus RNA vaccines too. So I wouldn't compare vaccine to vaccine, but one thing that RNA does is it doesn't have the potential to integrate into your genome. And remember that it's a very transient molecule, right? So it produces protein, and then at some point it's gone. It's degraded. So the chance of a long-lasting effect with an mRNA vaccine or an RNA injected into the cell is low because naturally it's an RNA that tends to degrade more easily. It's the reverse that's the problem. Keeping it there long enough to make a lot of protein, that is the challenge. One has to worry about that. And then, you know, it's not infectious on its own. It can be made to produce protein very easily. And it's a little less expensive than producing proteins in our day and age. So we'll discuss more of the challenges of mRNA vaccines in the panel. And this is a picture that needs no introduction. But all of these, maybe 5 decades of research then culminated in this award that we have, we are meeting for because of the award for mRNA vaccines and therapy. So this is essentially my last slide, where I'm trying to show that the true, so what one can achieve now scientifically and with technology is that you can have an RNA which can be delivered into the cell. Now it's up to our imagination what all we want to achieve from this technology. One thing, of course, is the obvious, which is to combat infectious diseases with things like vaccine, which is what we've talked about. That's just one thing that you can do. Because the technology is not only about vaccines. The technology is about delivering an RNA, which can then make a protein of your choice. So how about if these were to be delivered for diseases where you want to control proteins involved in the inflammatory response, right? For example, in cardiovascular diseases. What about using this kind of a technology to combat rare genetic disorders? There are several rare genetic disorders where it's the absence of a protein or a mutation in a protein. So a protein gone bad creates a life-altering situation. So in those cases, you could use the same technology, send in an RNA which makes that protein in its correct form, so that now that person has more of the correct protein rather than this mutated protein. So the technology itself of these RNAs is bigger and goes beyond vaccines. Okay, so I just want to end with saying thanks to the Tata Institute for Genetics and Society, where I work. And it's a motivating place where you're given a challenge. My team does not work on vaccines. We are only interested in them. We use RNA to solve some other problems. I'm happy to talk about them over tea. And there's a very happy and motivated bunch of young students that I get to work with. All of us completely fascinated by RNA. So I'm open to questions, I guess. Yeah. Thank you.
Good evening, Doctor. My question was regarding cancer, immunotherapy, and vaccines. So right now we're seeing an increased incidence of cancer cases, 1 in 2 men, 1 in 3 women. And I was wondering if there was any scope for preventive cancer vaccines versus the therapeutic cancer vaccines that we see right now. And also what are the roadblocks towards developing these kinds of preventive cancer vaccines?
So, in order to make something that's preventing, you would need to know some sort of a genetic basis. So there would be examples where you know of a mutation which can be corrected by a protein replacement kind of a therapy, and it could be used over there, and I think people are using it over there. But I mean, cancers are complex and go beyond that mutation. I think on the panel we might have a good discussion on this, so I'm just going to pass on the difficult questions for the panel, to be very honest. But
to make use of the mRNAs to prevent the transcription of proto-oncogenes, because that's where the cancer starts. So what would you say the process would look like?
So you're asking me if we can use RNA to prevent transcription of something.
Yeah.
Yeah, you would not use an mRNA in that case, right? So you would use, and there are RNA which are not mRNA, right? mRNA would make protein. Other RNA would simply go and function as RNA, which means it would bind to DNA, bind to RNA, and do these kinds of things. So you're absolutely right, and people are doing that. You can send an RNA. It does not have to be a messenger RNA. You can send that into the cell pretty much using the same kind of technology, and that would go and bind to whatever you want to block, to an RNA that you want to block for something like a proto-oncogene, and that would function as an oligonucleotide therapeutic. Yeah, and that's also being used. It's just that the nature of this talk was such that we talked about messenger RNAs, but the same technology can be used to block. Yeah.
Doctor, thank you for a fascinating talk. I'm a layperson, and in the interest of people who may watch this in the video later, I had 2 questions. So the first one is that you did talk about RNA and also messenger RNA, and you mentioned a tRNA. So if you could please just give us a little bit, summary description of how these things are different. My second question is that there was an important discovery, shortly after RNA was discovered, that a virus could actually be used to cap an RNA and produce proteins. Now, do the proteins, when the cap is put by a virus, change than the other proteins? So the part of what I'm trying to figure out here is, if you put a cap, you can get proteins, but if you change the nature of the cap, do you also change the nature of the proteins? Thank you very much.
Okay, so the first question is about the different types of RNA. Early on, after the mRNA, so there were 3 primary types of RNA which occupy our cell. There are many more, but I'll just talk about the major ones. One is the messenger RNA. As the name suggests, it converts DNA to protein. One is the ribosomal RNA, right? The ribosome is a machine which does this whole conversion, reading the mRNA and making protein. Something has to read the code, right? And then convert it into a protein. So the machine which reads that code and makes protein itself is an RNA machine. I mean, it's an RNA protein machine, but there's more RNA in it, so I'll call it an RNA machine. That's called the ribosome. So all the RNAs that are in the machine reading the mRNA, those are called ribosomal RNAs. They largely form a structural, it's like parts of a machine that hold it together. The third most prominent type of RNA in the cell is this tRNA or transfer RNA. The transfer RNA is also part of this whole convert mRNA into protein process. What it does is, now the protein has building blocks called amino acids. All of these are polymers. Polymer as in something gets repeated many times. So this repeating subunit called an amino acid, which makes our proteins. So the transfer RNA finds the amino acid in the cell and brings it to where this machine is. Because for this RNA to protein conversion, you need the machine, you need the mRNA code, and then you need the building blocks which are going to go into the protein. So even that bringing of the building blocks happens by RNA, right? So those are the three primary, because making protein is a very huge, it's a big process in the cell, and a lot of its resources go towards that. All three parts of this process are actually, involve RNA. Apart from this, there are many other RNAs. So, so the second question was about the viral RNA. And if the viral RNA is now taking a cap from the host and adding it to an mRNA, what will happen to the protein? So the cap doesn't, the cap is in a portion of the RNA which is not going to affect the protein that is made. It will affect how quickly the protein is made. It will affect how much of the protein is made. It will affect whether the protein is made or not, but it will not affect the actual sequence of the protein. That is somewhere else. So the part that codes for a protein is kept somewhere, and then the cap is somewhere else, close by, but not actually on that place. So the nature of the protein would not alter. But of course, if the cap will decide how quickly you make protein, and that may affect biology in many ways. You make too much of a bad protein, then you're in trouble. You make too less of a good protein, you're in trouble. So the cap is important for all of this, but the nature of the protein would not change.
Thank you so much for such a simplistic basic insight. I am a non-bio student, so as a layman perspective. You also mentioned about at TIGS, you have quite a young, vibrant team, researchers out there. So this question is more from a student point of view. Often, let's say 11th, 12th standard student going to college and planning for biology-based studies is like MBBS or don't know, right? So for a team of this kind who is into a research of this kind, what kind of skill, what kind of study background that aspiring student should think of to get into a research of this kind? If you can just talk, for example, the kind of young people that come with you for the research. So what kind of a study background they should come from? If that is a part of, if you can just briefly touch upon that, it'll be great.
So honestly, I don't know. I was a very bad student. And I also got really poor marks in molecular biology, particularly a field in which I now, live and breathe. So I don't know that, but I think any field, I think science, the way science works, is that it is really genuinely multidisciplinary. So you have a place for every kind of background because it has its own process and it makes you something through that process anyway. So I don't think it really matters whether you started as a physicist, a chemist, or a biologist. I don't think that background matters. I think some perseverance and motivation and general love for science, those are the kinds of, not disciplines, but general characteristics that would make for a good student, I guess.
Thank you, Dr. Arti, for that excellent lecture and the previous lecture. I'm a doctor, so I can ask you a few technical questions. The first question I had is, if you look at most of the mRNA vaccines, the antibody titers last for a very short time. That's why most of the Americans are taking their 5th dose and 6th dose of the mRNA vaccine. So the immune responses are primarily B-cell mediated in a mRNA vaccine. Whereas if you look at our conventional whole virion vaccines, like what we had in India, they stimulate a wide variety of protein antibodies and a T-cell response too, which leads to some memory. So how would you look at a kind of, when you have a choice between two vaccines, one which is going to give you a long-term protection with T-cell immunity and B-cell responses, whereas one where the immunity wanes very quickly and you keep needing repeated dose, which in our country is going to be logistically impossible. So these are the basic differences between these vaccines. So
Yes.
how do you think how the mRNA vaccine is going to fit into the public health program if you need to give repeated doses?
Yeah. So, so from what I know about the current state of mRNA vaccines, not the ones for COVID that were given that time, is that there are mRNA vaccines which are able to induce sort of, there's a fly over here, a long-term memory. So that pathway of immune response can be activated by mRNAs. It was not seen for the early vaccines that were administered, you're right, but I think there's research going on so that there is a titer that is maintained of antibodies, but also there is a memory aspect to the immunity that is induced. I think this is another question where maybe we will also discuss this in the panel again. But yeah, you're right that there is a trade-off. The trade-off is that it is a transient molecule. So while you get a response, right, and it's easy to manufacture, etc., and going to be much faster than conventional vaccines, the trade-off is that the nature of the RNA is such that as this, as the molecule degrades, at some point after that you will stop having a reaction, right, to it. But there is enough research going on now. So the mRNA vaccine is not a saturated area. There's a lot going on in it. One aspect of this is to amplify. So making RNA from RNA. There is an enzyme that can do that. So the self-amplifying vaccines are able to do that so that you continue to keep some amount of mRNA going in the cell. And other than that, yeah, in lighter vein, immunology was the other subject other than molecular biology, where I did very poorly. But maybe we'll ask this in the panel who work with mRNA.
Dr. Sanjay is going to be on the panel?
I don't think Dr. Sanjay is on the panel, but we do have a very good variety in panels.
Okay, fine.
Yeah, we will bring this up again though. We can discuss this. The half-life of different mRNAs is different, but about 30 minutes for some RNA in a cell. Yeah.
Thank you so much, Dr. Ramesh. Thank you so much, Dr. Ramesh. That was a lovely talk. And you had all the people listening to you very carefully. And we had a lot of questions, which means that the panel discussion is going to be shorter. But we would love to have all the questions that you had from Dr. Singh's talk or Dr. Ramesh's talk. For the panel discussion, I would like to welcome Dr. P. Chitra. Please come onto the stage as I introduce all of you. Dr. Rajesh V. Iyer, Tata Institute of Genetics and Society. Dr. P. Chitra, is an independent consultant in pathogen genomics. Dr. Sweta Raghavan, project scientist, dengue vaccine program at National Center for Biological Sciences. Dr. Mona Lisa Chatterjee, co-founder Sekhi Bio, housed at CCAM currently. And to moderate this session, we will have Dr. Aarti Ramesh, so you can direct all your questions there. I would request all the panelists to briefly introduce yourselves before we start the discussion. And again, to reiterate, we just have 1 hour. So we'll quickly go through the introductions and we'll take questions. And thank you so much.
We can start from here. Yeah. So as you mentioned, I'm Mona Lisa. In one line, I'm a product developer. I did my master's PhD in Indian Institute of Science, did my postdoc at Yale, but then converted to the fascinating field of drug discovery and development. 23 years doing it across metabolic diseases, infectious diseases, worked with small molecules, small chemical entities, novel biologics, and have been fortunate in my lifetime. A drug takes 15 years, reach market, reach patients, where you actually see the delight in you when you start something and somebody you know took it and actually. So I had products which have launched, gone into 60 countries, got approval of FDA. So seen the journey. It's a very humbling journey, but it's a very cross-functional journey. So that's me. And currently I've co-founded an innovation-driven discovery company called SekiBio. Thanks.
My introduction is going to be really, really short compared to Mona Lisa's. So I'm Shweta Raghavan. I'm basically a project scientist at NCBS. I work on a dengue vaccine program right now. I also have a Biotech Ignition grant that basically helps you go the entrepreneurship journey at CCamp. So this is me. And I work on an mRNA-based platform for dengue.
No, we can't do ladies first.
Yeah. I am Rajesh. I am a scientist at Tata Institute for Genetics and Society. So I was previously involved for making mRNA vaccine for Spike. We were in the turbulent times. But once that was over, now I have shifted my gears towards rare genetic disorders. And what we want to take, since both of our speakers has aptly explained to you that you can take the RNA and make protein inside the body. So we want to do the same for rare genetic disorder people where there is protein which is not there, use the RNA to make the protein inside the patient body. And hopefully it would be far less costlier than the protein therapeutics which many of the people are taking. Thanks.
Hi. Hi, I'm Chitra. I did my PhD from NCBS. My PhD supervisor is in the audience, Professor Sudhir Krishna. So we worked on human papillomaviruses and cervical cancer. And after that, I have been doing pathogen genomics, particularly in conditions of unknown etiology. I do sequencing, so I figure out what is the pathogen by looking at the genome of that pathogen. And so now mostly I work on emerging infections and how a virus, for example, is changing as it goes through the human population. Okay, thanks.
Thank you all. So I do have a list of questions, but I mean, in the interest of time, I'm wondering if we can just do questions from the audience first. Okay, I will happily pass that question on to the panel. So does anybody want to answer that?
So right now we are, so the way forward for any vaccine is that you go through a design and development stage. It needs to be tested for safety and efficacy. Once that is done, you go in for large-scale manufacturing, and then it goes to a whole clinical trial set, so phase 1, phase 2, phase 3 clinical trials, before it even hits human usage, right? Like widespread human immunizations happen. So we are still at the design development and preclinical study, where we are evaluating its risks, the safety, the immunogenicity. We really are hoping that we can take this forward. So we are in conversations with other entities, because this cannot happen at academic institute, because it requires people with various kinds of expertise to take the study forward. So we are in conversations with other entities, with industrial partners, including Genova itself, so that we can take this candidate forward, conduct non-human primate studies, and soon over the next few years, if everything goes well, we will soon get it out into the market. Thanks.
You have something to add?
I would just say in terms of other dengue vaccines that are already out there, right? Shweta, do you want to talk about it?
Yeah, so I was just talking about the mRNA vaccine. Right now, in the US, there is one dengue vaccine that's already approved. It's called Dengvaxia. It has conditional approval, and it's not widely used. People who take that vaccine needs to be seropositive, meaning they should already be exposed to dengue. In India, currently we do not have any vaccine that is approved. And most of the vaccines are mostly traditional vaccines. What does that mean? It means you take virus, you are attenuating the virus, meaning it is still live, but it is not infectious in that sense. And these are used as the vaccine. Now, what is happening with dengue is that, like COVID, dengue is not a simple pathogen, meaning it's not just one virus. There are 4 different serotypes that are there. So a vaccine, what does it need to do? It basically needs to protect whoever's getting immunized against all the 4 serotypes, right? But with dengue, there are additional complications, meaning that if I get exposed to one particular serotype of dengue now, and since in India, you have all dengue serotypes, and now we've just gone through a season of dengue, right? So what happens if I get exposed to another serotype? Things might not be very good for me, meaning whatever immune responses that my body generated the first time might not be sufficient to protect me against the second serotype infection. In fact, in many cases, it might promote viral replication. So these are challenges that are inherent of the dengue virus. So there are complications. So dengue has been around for more than a century, but some pathogens are just quite challenging to make a vaccine that protects uniformly across all the 4 serotypes and gives you long-lasting protection.
Okay, I have a question for anybody of you who want to answer it. So in the context of India, would it make sense to pick one kind of technology, whichever one, whether it is the traditional or whatever, and go with that and put in all the efforts in developing one kind of technology that you can really be a master of? Or would it make sense to bring on new technologies like mRNA, DNA? I mean, does it make sense? Is too much diversity going to be a problem in our context? Where I think we are still quite early in the, a little late in this game.
Yeah, so if we look at, so we heard a very beautiful, history of how mRNA has worked, the first generation technology, what pandemic did to it. But it is just the tip of the iceberg. The technology where very much India is in level playing field, and we hope from our space, all the people sitting here have IP. The second generation technologies, we heard one today, which was self-replicating. The second level is a circular RNA technology. In fact, Drew Weissman just rolled out a company last year, 2023, which raised about $270 million as Series A, one of the highest on circular RNA technology. The field is rapidly innovating. Why are they innovating? The first generation technology that you heard has a lot of limitation. It worked for COVID only with limited success. Many of the, you see, discussions came out because we didn't have time. You have to realize it was a balance.
So when you say first generation, you mean the
traditional linear capped poly-A. It does not have stability. It is immunogenic, whether we like it or not, it is true, right? It has reduced it. It has very short half-life. It does not make, it goes into the body, but does not stay there to make the protein for a long time. It is difficult to manufacture, scale, stable. So there's a whole scalability thing. Much better than proteins. You have to realize we improve from MABs. It's all relative. Yeah. So there is a real opportunity to take RNA and convert it now to many use cases. Now, I'll tell you one of the dreams that we have at SekiBio. There are many, many life-saving drugs that have been identified in the world as monoclonal antibodies. We have all heard about it. Innovative products, biosimilars that came, which brings down the cost. But if you look at the population, how much of our population can really access it? Because of the cost, the scale, the production. The large global populations just do not get it. Even in US, people don't get it. Let's be honest, US is not homogeneous with respect to access, right? So RNA actually has the potential to disrupt the entire biologic space, provided you have to adapt the technology to be able to make that much of protein in a relevant dosing regimen. You can't dose every half an hour. That's not practical to a patient, pardon me. And it has to be safe. Kindly understand, a therapeutic which is given chronically has to be safe. So the entire field is really gearing up. Similar for rare diseases. Today people are taking proteins which is exorbitant. Small children cannot get it. Please realize it's in our population, we don't give it. So you take it to RNA. Now why RNA is so attractive? If you can make if I just give simple examples, I'm not going to detail. Suppose 2,000 liter fermenter, anything that manufactures, the doses that you can make it is in 1 liter fermenter. The scale at which you can bring down the small footprint manufacturing, the rapidity, the rapid response, the cost of production, all of it makes RNA a really When you say India is late, I really strongly urge there is enough innovation going on in the field.
I said that just so I can get a response from
where
you.
it is not late. I will push back very seriously, it is not late. There is enough. We are actually today, the great thing is a level playing field. Many things are passed. CAP, IP, monopoly, license, we have to alternate it. So many things are not. But there are many things we can still innovate. Second thing is we have amazing scale-up power. If we collaborate with the right partners, you can actually scale up and manufacture, which we have seen repeatedly in India. So so that would be my response. Simple, yes. Allow the innovation, bring different technologies. Every technology will find its home to the right use case. As you rightly said, beautiful presentation. A silver bullet is not there. It's all to the use case. And she just explained how dengue has been traumatizing, in fact, right? So that's my response to you.
And you feel India is ready for all of those?
I really think India is ready for it. Now, this might be my optimism, but I really think it is.
Okay. And so, do you guys want to add to that or no? Not really.
No, just one point. So in terms of manufacturing, right, across the globe, India is one of the leading manufacturers for vaccines. And when you add multiple platforms to it, I don't think anything can stop us. Right now, Serum is one of the world's leading manufacturers, right? Like that, we also have Bharat Biotech. We have so many vaccine manufacturers. And now everybody is interested, especially if it's mRNA and with so many applications. Why are we hesitating? I think it's great, and the time is right. We have to innovate, move in that direction, and it doesn't hurt to have additional platforms.
So, Shweta, so I want to pull on that thread because you said, and I agree, that we have the capacity to manufacture, right? So then IP might be the real problem, right? Because what has to happen side by side? You have to be able to manufacture. You have to have that repeated technology, but then the new ideas for the next generation, where just making a little protein is not enough, it has to self-amplify, it has to be circular. Those are innovations that come from a research lab, right? So where are those, are we equipped for that? You know what I'm asking you, right?
I do.
Manufacturing is
so.
way down the line.
Correct.
If you want to do something where you're not threatened by or not restricted rather from the IP outside, that means that research has to go along side by side.
I think we are in the process. So unless we start somewhere, we will not know where the bottlenecks are. You're right. IP is a major hurdle, especially if we are going to be using the current formats of using self-amplifying or non-amplifying along with the LNP formulations. Many of them are behind IP firewalls.
So just to clarify, IP is intellectual property we're talking about, because if something is designed somewhere else and there is a patent on it, then it becomes restricted for us to use. Yeah, sorry.
So if you take the COVID vaccine, the mRNA vaccines itself, many of them are mRNA along with the lipid nanoparticle formulation, both of them, Moderna as well as the Pfizer-BioNTech. Now what is happening is that both of them are suing each other because of the IP, the patent infringements. And these are major challenges for anybody else to go forward. You can try to work between the cracks. You could probably think of, finding ways, loopholes, in these IP forms, but it also opens up new opportunities to innovate. And I think this is really great. I think since we've sort of built some base, some foundation we have, it's interesting to build upon it, get more learning, be innovative and creative. Yeah.
I'd like to add a life journey here, because this is something very close to our heart. So when we came back in 2029, we thought, okay, I came back from US and said that, okay, we're going to build RNA technology. We quickly realized we cannot democratize the technology. It is all under firewall of IP, licensing, infringement. We will never be able to get the cost which is meaningful to rest of the world populations. We said, okay, let's look at the newer technologies. Maybe they are IP free. The new technology, I'll give you a simple example because we live it day in and day out. So when we work with something called circular, where a linear RNA that you saw has just been 2 ends have been fused. Now, sounds so simple. Take the linear RNA, join it. That's it. Nothing else, right? There are, at least in last 2 years, 6 to 7 companies have opened. They have filed exactly the same methodology of circularizing. None of the IP is granted. They did grants. Whoever gets it, rest everybody gets excluded. You have to realize. So we said, okay, let's start from basic. First, find a new method of circularization. File your own IP. Second, find elements that will allow better translation on making protein. Third, entire LNPs are under IP. Right? The cost goes up. You have to realize anything that's IPs, cost goes up. You cannot give it to your populations. We said, okay, innovate on that. So I think bit by bit, we have to innovate so that we create something within India which is proprietary to us. And we are not blocked out from use. Sorry, I jumped the gun, but this is something which was really we live every day. Try to. Right.
I just want to know what are the advantages, disadvantages, and challenges in live attenuated vaccine and the recombinant DNA vaccine and the upcoming mRNA vaccine. Yeah. I just want to know about it.
We'll take that question. We just wanted Rajesh to finish saying, if he still remember, do you?
I remember that. So the patent landscape. So it is murky. The patent landscape has been murky, but India is the biosimilar capital of the world, right? We were never afraid of patents. We have taken so many cancer drugs and made it here. And Indian government as well as Indian courts have been very kind to people who have made biosimilars here, right? So entire point of patent is to stop you working. And if you do that, then ultimately you would be reliant on the West for all the time. So one has to take the leap of faith, regardless of the IP landscape. And as you go along that journey, you would always innovate and your own IPs would be created. None of the IPs which we are talking about, LNPs, have been coming out of air. They have been research of 20, 30 years of taxpayers' money, which has culminated into one patent. Similarly, if we are going on the track, so I think we should not be worried about patents. We should just keep on working on it. And if there is a patent which we are infringing, we should go for the government for the aid. Or if the end point is that, we have to license it. That is the only way to go about it. But it should not stop us from working.
I guess this echoes what was being said to some extent, that you move on at a certain pace with what you have, but then you have to invest enough in those innovations, because at some point, if you want to make it affordable down the line, it has to have originated here, right? Yeah.
Can I ask a very basic question here?
Yeah, just if you don't mind, there was a question, so we'll just finish that and then come to you. So if one of you want to answer that question about what are the very, not all the differences between all the types of vaccines, but some important things that you think we should all know about this kind versus that kind.
If somebody wants to start, you go ahead. I will follow.
Can I do this?
Yeah, you go ahead.
Yeah, so I can tell you a little bit about dengue because that's what we work on. And as I was mentioning dengue right now, in India, there are some trials going on, and they are based on live attenuated vaccines. Now, there are issues. For an mRNA vaccine, we use DNA as starting material. So we also understand that in some sense. So each one of them come with their pros and cons. So when I say live attenuated, what does that mean? It means I'm taking a virus, right? One kind of virus, and I'm attenuating it so that it is not infectious. And that virus, along with certain adjuvants or something, a formulation, I'm injecting it into a person, and that is my vaccine. Now, I have certain limitations, right? How many viruses can I inject? Now, for example, if you look at COVID, we have so many variants that are coming up. Every time a new variant comes, you cannot keep, indefinitely immunizing people, right? Over time, you might not have immune responses against those variants. So what do? You should be able to incorporate variant information. And how can you do that? One way to do it is look at nucleic acid-based vaccines that DNA and RNA are capable of doing. So I don't need to have 100 different viruses or pathogens. I can incorporate some of these information in DNA or mRNA and use it. I think Aarti did explain a little bit about DNA and mRNA. Where the DNA is located, it is inside the nucleus. So there's a membrane around the nucleus. And where is RNA located? Just outside that, just below the cell membrane. So when you're using vaccines, if it is a DNA vaccine, it needs to cross 2 membranes to go into the nucleus, whereas an mRNA vaccine needs to cross 1 barrier. So based on that, the amount that I would be using would vary greatly. So the amount of mRNA that I would use would be much lesser than a DNA. And for DNA to give rise to a protein, it needs to first generate RNA, and that RNA needs to produce protein. Whereas for an mRNA, I don't need to go back to a DNA. I can just directly produce a protein, right? So these are some differences.
I can add some very high level because I think that's what you're asking for. So let's take live attenuated. What is the biggest advantage? This is the classical way of making. We have often seen that immune response is good to live attenuated viruses. The challenge, viruses come with many different, biosafety levels. Imagine you're working in something which is extremely, pathogenic. You actually make an attenuation. It takes a lot of risk factor that's added. Second problem, which is a more serious problem, is live attenuated viruses have a low but a feasible risk of reverting. What you mean is, over many immunization, you may actually get a live virus back through recombination. So that risk stays with a live attenuated. Third is, as she mentioned, scaling making is not an easy with a live attenuated. If you go to protein subunit vaccines, very good. We have got beautiful fermentation technology to make protein subunit. But the thing is, the trick is to find the right immunogen. Somebody mentioned only adaptive response. What about cell-mediated response? What's the right immunogen to use? How much is it expressed? Then there is something called VLPs. That comes in with the manufacturing challenges. And you heard of RNA and DNA. So I think they all have their use case with the pros and the cons, with various disparate aspects.
Can I just make a comment, Aarti?
Yeah.
Your issue of IP, in the first 50 years, we were not a signatory to the global IP landscape. And so the flip side of it was that we could make cheap generics, but we could not innovate. In roughly 2005, we became a signatory to the international IP thing. Now you're in a situation where you want to innovate, but there's a lot of IP locks. One of the things that academia does not do is it does not lobby with the government about what you need. So one of the things, because by nature of the people who are doing the innovation, they're academia or industry-related academia. But given the fact that you have global markets, you have cost and you have need, one of the things is to tell the government that this is what the new IP landscape should be to protect your issues. And academia should step out of its comfort zone and deal with that.
Right. That's a good point.
Can we take one more question?
Yeah, there was a question over there.
Can you hear me?
Yes.
Mine is a very basic question because I'm not from this field at all. There's no connection. But talking about IPs and costs of vaccination, developing the vaccine, India during the COVID times developed these, we were the major manufacturer of vaccines in the world. And we gave it at levels which, or costs which, every person could bear. Now, how did India do that? If you can elaborate on that, if India was able to do that, and we are doing this in the space technology as well. So I don't know what's happening here, but we are able to do something in this country which is very different from these protective barriers which the Western world, if I can call it that, are actually putting barriers towards development of these things from a commercial perspective. So what is it that which can be done? And like the gentleman over there said, the idea is to just keep on innovating and researching rather than be limited by the IP kind of barriers.
I will go ahead.
Yeah,
Okay.
please.
Yeah. So with respect to the COVID landscape, the one thing which is abundant was the demand, right? That there were so many people afflicted. So the cost of the vaccine and all those technologies came to the most basal levels, because when you divide with the number, you can always get your profit. So that technology and the cost which is there for COVID is a very exceptional scenario, which will happen only in the pandemic cases. That part being said, with respect to, let's say, Covishield, in which we took the license from the AstraZeneca as well as the Oxford, and they gave us free license and we completely licensed the technology. And then, as she rightly pointed out, we have so much of a scale up and labor opportunities in India that we were able to make vaccine at a very low price. So that if you have to take this argument further, if we are making technologies in India, or if we are developing the skill set for it, regardless of whether we do an innovation or not, or whether we do an innovation later, we have the capacity to produce vaccines, therapeutics for the world at the most cheapest of the rates, because we are not working with respect to those constraints and those kind of high cost ratios which are there in West. With respect to India, with respect to the labor and the scale up stuff, we can produce vaccine at a cheaper rate. So I think in the case of Covishield, we were quite lucky that we got the license. In the case of Covaxin, Bharat Biotech got the strain and then they actually inactivated it. And that technology was there with us earlier also, because we were also quite fantabulous in eradicating polio and all those things. So we had the capacity to manufacture vaccines for a long time. It's just the COVID gave us an opportunity and we scaled up very nicely.
Rajesh, can I just follow up on what was said from behind and Rajesh? So Dr. M.K. Bhan, who developed the rotavirus vaccine, he set the price of the rotavirus vaccine as $1, if I remember correctly. So you have an opportunity to tell the government, and governments also want very simple signals. If you say, Moderna, Pfizer, whatever, the cost of mRNA vaccine is very high, hundreds of dollars or thousands of dollars. If you say that, you talked about the time limit for making a vaccine. If you say that as a community, you will make the mRNA vaccine available globally at this cost, that's a simple message for the government to process and to put all the steps through. And Dr. M.K. Bhan's rotavirus vaccine of I will do it under $1 was a very good example which you already have. And it covers the point at the back, which is a version of the space program, which is done at 100 the cost that the West does.
And I must say, when we innovate, we actually we have put 50 micrograms $1. Today, we are 50 micrograms $5 without a scale up. We are at the lab scale. So, as you scale up, you can actually bring down the cost. But I completely agree. You need, like you have target product profiles for scientific attributes, you need a cost attribute, absolutely. Right. And work backwards to that.
Yeah, ma'am, I have one question. One of you were telling that spike mRNA technology is better than the earlier live attenuated virus. Can you hear me? Yeah.
Yes, yes.
I'm sure that you all have heard about Sucharit Bhakti, Peter McCullough, Stephen Lanka and all those people who have told the dangers of mRNA technology compared to live virus technology earlier. Because once the spike proteins start getting generated in the body, nobody can stop it. You have not put a off button. You have just put the on button. So are we all becoming lab rats? Because God has, I believe in there is some Almighty who has created us to live for some years. But are we, with all these injections, are we putting a stop on our life, like curtailing our life? Because we don't know, like anytime anything occurs, these spike proteins will start attacking it. And usually autoimmune, most of the thing is because they think that any kind of attack happens, even they might start, the cells will start attacking themselves, thinking that it's from outside body. I mean, that is how it's programmed. So what happens is, body will start attacking itself, and we have already seen the result, autoimmune diseases have increased. So what kind of...
Since this was the same question that was asked to Dr. Sanjay, maybe we will try another question
Yeah,
right
because
now.
we should not risk our life in future. We are also, I am also in that. So I
Yeah. So one of them, for example, is that the spike protein can't be switched off and it goes on. So maybe a little bit of clarification, because on one hand we've heard from you that mRNA is extremely temporary, and that's the problem, that your immune response dies out. So the two can't be true. So it has to be, something has to be what is established with the conventional science today. Maybe if the learned people can share it, that would be useful.
Okay, so that's a good point.
I can give you a view and we can discuss, and we fully respect. So if you look at, when you're putting a live attenuated virus, imagine that risk. The virus is there. You know how it is made. It is passaged means grown, till some change happens, which often is also not characterized, by the way, in its genetic material, that it does not stay as virulent, is the terminology, means as potent, right? You are putting that in your body, which has all the spike protein, right? Which is then, of course, virus has spike protein. You are getting the spike from the virus itself. What is spike? Spike is actually decorating the virus. It's on the outside of the virus. Now the spike goes and binds to a specific receptor, which is host receptor. Receptor means another protein. It is hijacking our system. It is smart. Right? If you look at SARS-CoV-2, it binds to a receptor called ACE2. Okay? Now, ACE2 has a lot of activity, real activity, physiological activity in cardiovascular. So, a lot of cardiac side effects that you see, maybe just because you are integrating, that is biology, we can't change. Virus has become smart and taken that receptor. Now, whether you take live virus or you take spike is a part of the live virus. I hope I'm making that clear, right? So even when you take spike protein, as she mentioned, the RNA you put, right? RNA get taken up by certain cells in our body, which makes the spike protein. It gets chopped up. It gets presented by what is called an antigen presenting cell. Looking at that, our machinery, B cell, with the T cell help, sorry if I'm making it complicated, now gets activated and makes antibody. Right? That antibody sits with you till the real virus comes. Now, real virus has the spike. Now, why are people saying when the variant is coming, our vaccine is not working? Because the variant is smart. It has changed its spike. Receptor binding domain has got changed. So the antibody that is generated now cannot attack that various, right? So if you see, the spike that is generated also has its half-life. Body will chop it off. It does not stay ever and ever again. Actually, the linear form of the RNA is very short-lived. The half-life is coming from the spike protein itself, which then get presented. The major challenge that has come from the spike is because of the ACE2 engagement, because ACE2 has a lot of physiological functions. I hope that's one perspective I can share. It's a perspective, but that's one perspective I can share with you. I think others should share. This is a very,
it's important to discuss.
It's important thing to discuss.
Okay, yeah. So, with respect to mRNA vaccines, the risk which you are talking about, autoimmunity, is associated with any vaccine, right? This vaccine, we are being saved from smallpox. I don't know, we eradicated smallpox. And all of these things, they are from the vaccine technology, right? So it is not that we are being not taking, if you take up one specific example of a vaccine, it becomes completely out of context. We are taking vaccine for now 200 years. So at the same point, we have not seen any kind of increase in autoimmunity. Yes, I'm coming back to you. So nowadays, if you are seeing there is an increase in autoimmune disorders and everything, because we as people are also progressing. There are discovery of new technologies, new diagnostics. There are new screening methods. The doctors are more informed. In the earlier times when autoimmunity was not even considered, now the doctors consider it. So those cases now come up. It doesn't mean there is an actual direct correlation between vaccination or of these diseases. This has been found not only by, in India, CDC, WHO, everybody recommends there is no direct correlation between vaccination or any other disease or any other deaths. It's actually counterproductive. If you don't take vaccine, there is more deaths. So in a way, I understand there is no technology in world which is out of, which doesn't have anything, no side effects. You have to accept it. But in terms of vaccine has protected us for 200 years. Think about next influenza pandemic or anything. We need vaccines. And for that, we have to be prepared. And that is the most important thing.
Do you want to add?
That is the worry, because before launch, like, it should not happen that we will see like mass deaths happening and WHO tells there is no relation, because we know who is funding WHO. Everything we know.
So maybe I can just comment on this. So first, yes, it is true. This is a new technology. But this technology has been for the last 3 decades, as we heard. People have been researching on this. And let me just, thank you. So basically, so you're right. It is new for the world at this kind of scale. There were vaccines that were already very close to approval. So there was an Ebola vaccine that's in the process, Zika. There's a CMV vaccine that's now in phase 3. It's an mRNA vaccine. RSV vaccine as well. So there are quite a few that are in the pipeline. And for the mRNA vaccines that are there, other than India and China, most countries in the world have received the mRNA vaccine. So now we have a lot of people have already received this, and they have been studied, there have been trials. There's also reporting of adverse incidents. So we will know more, and we are knowing more. I think there are like billions of doses of this vaccine that has been given out, because there's a vaccine, then there have been boosters. So now we have enough evidence, and I don't think we are in that place anymore where we need to have those kind of concerns. So this is now a well-documented problem. We can go look at the results of all these trials. Maybe that's...
there was a question. Okay.
I have a question for Mona Lisa. Apart from IP, is there any other barriers for vaccine manufacturing in India, like know-how or...
No, you are generalizing. Okay, vaccine manufacturing has been always happening in India. You heard
I meant mRNA vaccine.
Okay, perfect. Again, mRNA vaccine, it depends. What do you mean by manufacturing? Are you going to be a contract manufacturer who in-licenses a product? You just heard Genova. So there are manufacturing. So many models work. There are partners who partner with the innovator company. Manufacturing happens in India because of our real efficiencies and quality and the scale at which we can do and the cost. And then it goes back. It's a contract with the innovator who then decides where it goes, at what pricing, etc. That's one model. Nobody prevents you from doing it, because it's a relationship between a manufacturer and an innovator company. That's one, right? The second kind of manufacturing we are saying is in-country developed vaccine, where you are not in-licensing the technology. What happens is, suppose the second modality is I'm a company, I in-license a technology, I develop the whole product, commercialize it, but then I have to pay licensing fee to the technology provider. Does that make sense? It's simple, right? What does is then you pass on the cost to your patient pool or customer. Simple, right? So that's the second tier. All are good. Please don't, I'm not saying this is better or that is better. I'm just saying each has a place. The third is you actually innovate in your own country. Innovate with respect to platform technologies.
innovate with respect to molecules. There are different ways of innovating. Innovate with respect to formulation, which changes, which then gives you freedom to operate, that you can decide the pricing modes. Okay? Again, you may choose a very different model, please, commercial and science. But what I'm saying is it gives you a space, what that gentleman at the back says. Why are they able to bring down, make it frugal? That's when the real opportunity is. That's when you can make it at costs that start making sense to our populations. So when I say, is IP the barrier? Manufacturing, no barrier. All the big manufacturers partner, and you should partner. If we have certain strength, why should we not make it? We have made a huge difference in the epidemic, you know. But we have to also go up on the value chain slowly, no? So that we can make a moon kind story and not be satisfied only being the manufacturer. That's all I meant. Please don't take it otherwise. Yeah.
I have a more general question, not restricted to vaccines. And that is on Saturday, 3 days ago. Someone had a stroke, someone that I know had a stroke, and had to be given what's popularly called a clot buster. And this was given not in one of those fancy corporate hospitals, but at Nimhans. It cost nearly 40,000 rupees. Incidentally, it's made by Genova, tenectase. This is obviously an unaffordable level for most people in India. You wouldn't go to a hospital like Nimhans, even if you're living in Bangalore, you can get to Nimhans quickly. You can't do that elsewhere. So somewhere these advantages that we notionally haven't been exercised because this tenectase has been commercially available at least for 25 years. So it's not as though it's something new where you're being gouged for the IP. At the same level, I wonder whether extending Aarti's question or re-asking that question, I believe what she asked was, if there are so many technologies available, should we prioritize? Can we afford to put money and other resources into all these things? Intellectual resources, physical resources, whatever else you want to call them. That doesn't seem to be an answer, at least just now.
Because I answered that, I'll give you my perspective on it. I really believe there is a place for, yeah, so I didn't answer. So there is a place for each player. I also incidentally was the person who started biosimilar programs in Biocon. I've spent many years there. And also the whole program about insulin, where, as you know, once it was launched, Novo dropped its price to 1/4 of the price, right? So all the stories that you're saying. So there is a space for innovation. And I completely agree with you. There are many life-saving drugs which are not getting available. There is a place for biosimilars. There is a place for generics. But if you say, and this is my perspective very respectfully, we will, for today, if we only fund the generics industry, which is actually quite well established in India, and biosimilars industry, and we will prioritize few innovations, what it does is, again, my perspective, it takes India back. We cannot come up as leaders in some of the stories. Again, as I said, this is, I'm giving, because I've lived a small molecules, a biosimilars, and they were wonderful journeys where we could bring down the costs.
I'm not disagreeing. I'm just asking if we have the resources to distribute across all the things that, let's say, NIH can do.
That's a government policy question.
Right. So maybe I'll address two things that you said. One is about healthcare and the cost. And I think part of the answer to that might be economic models of how the fact is that I think we have like 70% out-of-pocket expenditure for healthcare in India. Even more. Yeah. So we have that challenge. And there are ways to address that, because you also have to incentivize companies to do innovative products, and they are expensive. I don't know about this particular product, but I know that companies invest a lot and they would like recovery of that. And to incentivize that, you cannot necessarily bring down costs everywhere all the time. So that's one point, that we might have to think about different models for when a person gets that molecule, right? And insurance is one way, and having the government cover it a bit is the other. The second part that should we be investing in innovation and across? Most funders will do a prioritization of what they fund. And the one thing that I can tell you as a researcher who works on technologies that can't yet be brought to the bedside, in a sense, is that we need to have some in the bleeding edge, where we lose more money, it is more expensive to innovate. However, I think it is like, I think we are sort of poising to be lucky.
I wasn't asking if we need to innovate. What I was asking is, can we afford to innovate across the spectrum?
Right, right. So I agree. I think that we have to prioritize. However, we know from experiences like the way the mRNA vaccine was developed, some of these researchers actually didn't get funding. One of them actually couldn't stay in her, didn't have a position. So it tells us that it's hard to know what's going to work. So that's why it's good to spread. At the same time, you don't have the resources to spread.
I want to add a point here. Yeah. Right. So when you look at these, so there is a very great impetus that you need to divest. We need to work on many technologies. And the best answer is that if we have monopoly there, always the cost would always be high, because these industries, they are working for profit. So when we have, let's say, there are 3 companies who are working on a protein-based therapeutic, then third company is working on an adenoviral-based therapeutic, or something is working on mRNA, then a competition arises where the customers are going to get a price advantage, just like the mobile industry has given us. So I completely agree with you that there has to be a continuous impetus in multiple technologies, because we don't know which one is going to be performed the best. We can all be, because I work on mRNA, I would be very optimistic about it, but at the same time, there might be a protein guy who is very optimistic about it, and all of them have to be together.
And you must be willing to accept failure.
Absolutely.
Obviously.
Absolutely. And you need
No, actually build on them. Actually to build on them.
And you need to fail early so that you burn not all the money and then fail. That's the...
I know the time is short and there'll be other questions, so just take the question and respond to it. mRNA vaccine in COVID actually had one problem, was the physical logistics, that you had to store it at very low temperatures that were not available in this country. Is that a characteristic only of COVID, or is it a characteristic of mRNA vaccines? Don't wait for an answer now, when you discuss later.
Is it fair to say that was then, and now there's enough technology to have solved that problem? So maybe should we take one last question?
Okay. Yeah, last question, or mine is one?
No, yours is not the last currently.
Oh, somebody's asking. Yeah, go ahead.
No, please go ahead. Please go ahead.
Yeah, so there were a couple of quick questions. One is that, during the COVID time, there was huge cooperation, right, across the world. Is it continuing? That's one question, or it has stopped? Second was, we heard a lot about this Wuhan Category 4 lab, right? So there are some, I think, 20 or whatever across the world. How many we really have in India? The third is more of a thought. See, I don't understand this space much, but I do understand a little bit of technology. And what I've seen is that this same discussion of, we can do everything and we have it and nothing is a problem, I heard in technology space, but ultimately, we lost the product opportunity. Now we are losing the AI opportunity, right? So the thought will be that, while ambition is great, but, people like you probably should think about, sit together and really create something which is a sort of a document and publish it so that, we don't lose yet another opportunity, just like we lost many other opportunities, right? We have some advantages, which you talked about, right? So those are the couple of questions and comments.
Okay, so the first, sorry, I remember the second question. I don't remember the first. The collaboration. I'll pass the question to you.
Collaboration is one.
Labs.
Second is there to thought about some super vaccine for all these SARS and MARS and COVID and common cold. Is it happening? Because
it's happening.
Yesterday we have been told to take some new vaccine. Pneumonia we take, and all we are very confused. Third is what sort of lab facility do we really have?
Yeah.
The level 4 and all that. And fourth is just a thought that you can put a document together so that can you follow. Otherwise, 20 years on the line, we will think that we missed yet another bus.
Yes. So, do you think the cooperation that the world saw during the pandemic is still
Widespread.
is still on and widespread?
I think the WHO model for mRNA vaccine is a thing that it's still working. The hub and spoke model, where they are trying to develop an mRNA vaccine platform across India. Sanjay said that he's part of our WHO. There is entire of the WHO is setting up an mRNA vaccine completely industry, which is going to be indigenous, and all those patents that they are taking care of. It's called as an African corporation, which is there, which is going to operate in Africa. So there are lessons which are there, and the cooperation always goes about. But technologies as such, the most bleeding edge technologies it takes time to get democratized, right? So it would take time. But at the same time, the cooperation still exists. I don't think so that we have forgotten pandemic. Maybe give it 5 years later, maybe we would have forgotten about it. But right now we haven't.
So in order to add to that, when you say cooperation, is it cross-border cooperation or within the country?
What happened during COVID, right?
Correct. So there were a lot of these, from CEPI to WHO, which came together. You had that entity for vaccine distribution across the globe, making vaccines at reduced cost. All of those were there. But that was also for an event like COVID, right? This is not going to happen very often. But from there have stemmed certain other activities. Like, for example, CEPI has come up with these concepts of a mission for making vaccines in 100 days. They want to create a library of various vaccines that would be very easy to scale at a very short time. So these things are happening, and this encourages collaboration, and it is not restricted to certain countries only. So I think even Dr. Sanjay Singh mentioned that he has received a CEPI grant, and it has a presence globally. So these things will continue to happen, might not be at that scale, but it's not going to disappear.
So then,
Sorry, go ahead. I can take the second question. Pan-family vaccines are being developed. NIH had a big focus on, instead of every season, if you say influenza, can you make universal influenza vaccine? There are efforts to make COVID, RSV, influenza together, pan-flavi, right? So it is an effort that's ongoing, not yet into the market. But there is a very big thought that can we cover one vaccine for entire respiratory or the entire family? Very much there. As far as I know, you guys may know better. I know there are 2 BSL-4 facility in the country. I don't know, you might know better.
Yeah. The NIV and Bhopal for animal. Yeah.
So it's a little bit too limited.
But I think during the pandemic, the government of India also sent out, calls for BSL-3 facilities to come up, and many of them have come up and they are functional now. So I think in terms of infrastructure, we have gone a level higher.
So this point that you have to be aspirational about the policy is very important for us. Because you're academics, private, government, so he talked about a document, but be aspirational about a policy. I'm just repeating myself. Just be aspirational about a policy structure which drives your program. Not just institution oriented, but broader.
I think we will have one last question.
okay,
I just have one question.
Okay.
Very technical question. Like, what happens if you use both pseudo UTP and methyl CTP as modified nucleosides?
Okay, that can't be the last question.
I mean, I think you evaded questions, so I'm going to put Aarti under the bus, but yeah.
Okay,
So,
who wants to take that last
okay,
question?
I will take
no,
that.
that's not the last question. We'll have one more after that.
Yeah.
Come on.
Okay, so
I just want to know the preservatives.
No, wait, sorry, he'll just answer that question.
Okay, so as everybody, the Nobel Prize was given for modification of the RNA, right? So RNA is highly immunostimulatory. They were always plans to use RNA as vaccine, therapeutics, but the problem was when you give RNA, as Aarti pointed out, there's a sudden immune reaction, right? And then to what Katalin Karikó and Weissman did is that modified it. So now you're talking about modified bases, right? So they are, RNA is composed of A, T, G, and, sorry, A, C, G, U, right? So in this case, there are two nucleotides which are called as pyrimidines, that is cytosine and uracil, right? So in the case of mRNA vaccine, the entire of the uracil was changed to 1-methyl pseudouridine. What it offered it made RNA a little bit immunologically silent, and it helped the RNA vaccine to work better, and that's the Nobel Prize. Now you're talking about whether we can modify cytosine. Yes, people have modified cytosine. There are now people modifying adenine also, and there are people modifying guanine also, right? Now, it has to be a cost to benefit ratio, what you want to do. Because every time when you add a modified nucleotide, you are looking at a different kind of production process. If adding in the case of vaccine, 1-methyl pseudouridine addition was quite beneficial. But if you go for the next vaccine, and in that scenario, 1-methyl pseudouridine is working same as 5-methyl satidine, no point of going about it. But if both in combination are working better, so it's empirical. So one has to work, and that's what preclinical as well as phase 1 clinical are going to tell us. But we don't have a kind of a rule book here. You do change one and it will work that way. One has to work on it.
I just want to know the preservatives used in vaccine and its effect on health.
I think there's the way it is. I think you can take it. Okay.
You want to take it? You can take it.
So different many of these they go through a phase 1 trial, phase 2 trial, and there's a great amount of toxicity which is done in rats, rabbits, and everything, right? So it is no way that it has been taken very lightly before going about it. Most of the ingredients which are there, and let's say in a Moderna vaccine, there is some sucrose, there is some kind of tris, these are all considered and generally regarded as safe chemicals.
And all of the entire composition is tested for toxicity.
Entire composition is tested for toxicity. So in that scenario, we try to make sure that we are not giving something toxic, right? So that is kind of an...
So I'll give you a little bit more, It's
made
It is made public. Not for Covaxin, but most vaccines.
Even in phase 2 also they give the data. Because the problem is that everything,
Yeah, let me just, you're mixing two things, pardon me, okay? What you're asking when you say preservative, you're asking the entire formulation. The whole field is a formulation sciences, right? The tablet we take, the solution we inject, one is called API, which is your active ingredient, which is your RNA. You club it with the formulation, which will take all your lipids, but everything else that is there, which is true for every medication. Please understand, when we take a tablet, a syrup, there is a formulation that goes where the active ingredient is going. As he mentioned, when you finalize your preclinical candidate and you have fixed all your safety studies, your preclinical talks, as well as your clinical talks, happens in that context of the formulation. Am I making sense? So the safety package is with the formulation, is not for API alone. That's the first step. Second thing is, when formulation scientists develop formulations, there is something called generally safe ingredients. They often try to predominate it with that. The FDA or all regulatory body comes up with a list. Having said that, I am not saying that formulation cannot have a safety impact. In fact, LNPs have been known to have its own, especially the cationic lipids, have been known to have its own safety, risk. But you have to remember, when we take any dose, you have to look at safety, how development happens. You always have to show safety X over the given dose. I don't know if it's making sense. If my dose is 10, what is my safety window before I start seeing adverse effect? The question you were mentioning is, yes, in COVID times, it was an emergency approval. Pandemic did a lot of good, but pandemic also brought the urgency that some of the risk we had to accept. But remember, they never got full approval. They have to show the entire regulatory path to get actually the full approval. So while it is true, we did not shortcut the regulatory safety assessment. The only thing that had to be done is, it was a life and death situation, right? So I'm assuming
the regulators, I'm not a regulator, had to weigh the risk versus reward situation to say, somebody brought up a duration of protection question. It is true, we accepted the vaccine without testing duration of protection. It is not the classical development of a vaccine, that is why it takes so long for it to come to the market. So there are some pros and cons we had to do in the COVID situation. But that doesn't mean that all companies could just, scot-free and didn't have to show it. They had to build the regulatory package. And I will really urge you, a lot of this data actually is available publicly. It has been made public. All the formulation data by mandate has to be made public in the BLAs. Or, you cannot not disclose it and get it approved. Doesn't work like that. It will be available. All your safety data gets published as publications. Pfizer, Moderna, all of them have put it. I think Genova, didn't Dr. Singh talk about a couple of publications that, and they sit with the regulatory bodies, often can be accessed.
FDA data sheet which
FDA
comes with
data
it,
sheet.
which is
So maybe we can take this outside. Can we?
Thank you so much. Ma'am, we can take some of these questions later. But in the interest of time, we will have to really conclude this round. And thank you everyone here who's taken the time to come here. Thank you for asking those questions. That is something that we were anyway trying to address through these panel discussions and talks. Thank you, our panelists, and Dr. Aarti Ramesh for doing this.